that. It's wonderful for me to be able to introduce Rosanne. I asked Rosanne what I should say, and she said, oh, not much, it's short. But I think that it's only fair to give a little bit of Rosanne's background. She began her mycological work in Iowa, working with Lois Tiffany, and then her PhD at the University of Minnesota. I was lucky enough that she came to Harvard on a couple occasions, but for a couple of years where she was a postdoc, and she taught the biology of fungi course one year when I wasn't available to do it. So Rosanne has been a kind of mainstay in our lab, as well as in the lab in Florida, where she is now as a research scientist. Rosanne works on the Pezizales, or the Pezizomycetes, as we talk about them these days. These are the operculate discomycetes, and these happen to be the area where I spent most of my life working and doing research. What Rosanne and others, but Rosanne particularly, has done is to point out the biology of some of these fungi, particularly endophytes. I'm sure she'll talk about that a bit. I will say that she is the best collector I have ever been in the field with. She is fantastic, not just digging up these truffles down under the ground, but she sees things that the rest of us don't see. So it's a joy always to be in the field with Rosanne. I don't know that I can say a lot more other than I am just delighted to be introducing Rosanne, and I'm looking forward to her talk. So Rosanne, from the University of Florida, take it away.
Don, thank you for that really gracious introduction. I'm not sure that I deserved all that, but thank you so much. And thank you for inviting me to give this talk. I am really happy to get to talk about one of Don's favorite groups of fungi. So I'm a little bit nervous having him hear it, but here we go. Let me go ahead and share my screen. All right. Right, you all can see that?
Yes.
Dishing on the cup fungi, the Pezizomycetes.
Looking great.
Okay, so first I'd like to do a little overview of this class before talking about my own research. This class is named for the first fungus described in it, Pezizales. They're cup or saucer-shaped fungi with or without a stalk, and most have apothecia, or a modified version of an apothecium. We'll talk about that at length. The most well-known cup fungi are the morels and the truffles, which are also the most pricey and eminently edible. This order also includes some charismatic members, such as the fairy goblets. Let's see if I can point these out. The fairy goblets, the eyelash cups, and the saddle fungi. The highest diversity of ascomycete truffles belong to this order, and that's how I got into studying them. Let's see. How do I advance this? Oh, here we go. Sorry. All right. So the Pezizomycetes are made up of a single order, the Pezizales. The asci are unitunicate and operculate, which means that the walls are single, and they open at the tip with a lid. At maturity, the spores are shot out. The ascomata are apothecial, except in truffles and a few members with closed fruit bodies. And there's currently about 22 families accepted, about 200 genera, and about 2,000 species. You need a microscope to view some of the characteristics, which include an ascus that is amyloid, for Pezizales at least. And in this case, that means that they have a reaction in an iodine solution usually we use Melzer's, and that reaction can vary from just being at the tip to being diffusely all the way down the ascus. And this can have some phylogenetic significance. The ascospores are typically colorless, but some are purple, brown, or yellow. And the ascospores may be smooth or ornamented in globose to ovoid to more or less elongated. The ascospores are single-celled, which is one way that you can distinguish them from the Leotiomycetes. The size varies from less than 300 micrometers. So you have to see it with a dissecting microscope, to specimens of Gyrometra, such as caroliniana, that can be bigger than 12 centimeters across and can weigh 7 pounds or more. The fruit body is not too complicated, at least for the basic structure. It's composed of an excipulum and a layer of, or hymeneum, of asci and paraphyses. That's what an apothecium is. But the apothecium can be modified to a great extent. So here we have Ascobolus, which is pretty simple. It's just asci that are being produced at the top of a cushion-shaped structure. Povinula just has a kind of a saucer-shaped structure. Sarcosypha has more or less cup-shaped structure. Urnula is actually pretty urn-shaped. Then you get into the helvellas that are saddle-shaped, the gyrometras that take the helvella shape even further, and then morels that have in-pocketing of their apothecia. There are several main lifestyles. Squidolinia, microstoma, and Ascobolus. We think all of those are saprobes. That means that they're getting their nutrition from dead plant material. Here we go. Also, we have ectomycorrhizal Pezizomycetes. I would say the saprobes are probably the most common, but there's a great number of ectomycorrhizal ones in the group as well. And just as a short course in ectomycorrhizae or mycorrhizae in general, there are two main types. There's a number of different subtypes, but the arbuscular ones you can't see with your naked eye. You have to use a special staining technique. And that is, that's a fungus that is not among the ones that we're studying. And it is a relationship that is the most common among mycorrhizal relationships. The ectomycorrhizae, however, are common in dominant trees such as oaks, beech, all of the Pinaceae. Those are obligately mycorrhizal. They can't live in nature without their ectomycorrhizal fungal partners. And in turn, the ectomycorrhizal fungal partners can't live without their host trees. And it's a symbiotic relationship, mutually beneficial, in that the fungi are bringing in nutrients and water and offer some mechanical protection, and they're getting carbohydrates from the tree. Then we have a smaller group that are parasites. Most of the parasites are fairly weak parasites. This is Calicypha, which has been implicated in conifer seed disease. Octospora and its relatives that are moss parasites. And then Don's group worked out the parasitism of Winia americana on Armillaria species, which is pretty exciting. There's only one truly creepy Pezizomycete, in my opinion at least, and that is Phytotrachopsis. There's only one species, omnivora. It's known as the cotton root rot. There's no fruit body known, and it's a very serious pathogen that infects more than 1,800 host plants. It survives in soil as sclerotia. And I'm not going to mention it further, but it's a good segue into a story I have about what I studied as a graduate student. Because I was when I was first started my doctoral program, I came out to the Farlow on a visiting scholar fellowship to visit with Don Feaster and Matthew Smith, who was a postdoc there at the time. I was trying to decide whether to pursue a study of the ectomycorrhizae of Pachyplodes, which is a truffle, or the possible anamorphs of Pachyplodes, which are mitotically produced spore mats. So they're clones. They're not produced sexually. They convinced me to go after the spore mat research, specifically to molecularly link the spore mats we see on soils in the woods to their fruiting bodies, which usually occur at different times in nature and are not culturable. So we really didn't know what they were. This was a little scary to me because I was afraid, or I was imagining getting lots of non-target species like Aspergillus and Penicillium, and maybe just putting a lot of energy and dollars into not getting any good results. But they convinced me. And the reason that I wanted to study this was that there had been a spore mat that was implicated as a relative of Pachyplodes. Its name was Gliscoderma, and it had been collected by Korff in the New York woodlands. Here's a picture of one of Korff's collections here with the beech nut husk. However, the Gliscoderma that had been described from Europe, and it had been described with a real peridium, whereas these things that Korff had found were fluffy, but they weren't an actual covering structure. Gregoire Hennebert, who was an associate of Korff's, untangled to a certain extent the botrytis-like Pezizales anamorphs, and I used these to help me decide what I was picking up, whether what I was picking up was a Pezizales anamorph or not. And the Pachyplodes ones were pretty distinctive because they had a coralloid structure, and most of them had spores that were ornamented. So how did I go about this? Well, this is an alignment of two different regions, but every row here is of the same species. This is the ITS region on the left and the LSU, or large subunit of the ribosome, on the right. And you can look at the LSU alignment and see that there isn't a lot of difference among any of the things that I've aligned. And that's why it doesn't make a real good delimiting region of the DNA. The ITS, however, you can see lots of variability, and that is exactly why it was chosen as a barcode for the fungi. So this is what I used the ITS region to match up the spore mats that I was collecting with their fruit body. And it worked. So on the left, we have the spore mats, and on the right, we have the fruit bodies that they matched. Almost all of the spore mats that I collected on soil and sometimes on rocks and down leaves and sticks were Pezizales. Many were in the genus I was studying. These were particularly colorful and easy to see. We also found some spore mats that didn't have any fruit body that we know of. These included a Chromelosporium-like fruit body, spore mat, and a Chromelosporiopsis. These are also present in, the Chromelosporium-like one is also present in Europe and Asia. The Chromelosporiopsis, we know that there's at least one in Europe and at least 2 species here in North America. We found some that were previously unknown, like the spore mats of Hydnobolides. This was one of the few that we found at the same time and place as its fruit body. Here's the truffle down here in the right-hand corner. But generally, the fruit body was only there when the spore mat was fruiting massively. Those were all Pezizales spore mats. The non-Pezizales spore mats like tuber, the Morchelaceae, and the Dissinaceae were less easy to spot. But once you got the search image, they're not that hard to find. In fact, during one of our forays, we stayed in a place in the woods where the stairs to the apartment we were staying in was covered with tuber spore mat. Seemed auspicious. While I was working on my doctorate in Minnesota, I received some collections of a fungus identified as Marcellina by the collector. I was really excited about it because it's one of the few cup-like fungi that's related to Hydnobolides, and I had never seen it before. So I wanted to sequence it, and I did, but the ITS, when I blasted it, just came up with a page full of endophytes labeled as Pezizomycete endophyte. And that didn't really tell me anything. So if I had given it to Don, he would have known it right away, I'm sure. But this was while I was still in Minnesota, and I also wanted to figure it out myself. So after sequencing the LSU, I could see that it was a Plicaria. And after a bit more work, I figured out it was Plicaria endocarpoides. But I wondered how many other people have had this experience of blasting a sequence and then getting only ectomycorrhizal root tips or endophytes. It's a little bit daunting. It doesn't really help you figure out what you have if that was your purpose. So when the opportunity came up during COVID lockdown, Matt got in contact with a person who has done more work on endophytes than any other person I know. And that's Dr. Elizabeth Arnold, who's at the University of Arizona. So I've told you that there are three main trophic guilds in the Pezizomycetes: saprobe, ectomycorrhizal, and parasite. But it turns out that a lot of Pezizomycetes spend a portion of their life hanging out as endophytes. Matt Smith, Dawn, and I decided to collaborate with Betsy, who has done a massive amount of work on endophytes all over the world, to figure out which Pezizomycetes were the most common among endophytes and which were perhaps accidental. Many Pezizomycetes are endophytes in angiosperms, gymnosperms, ferns, lycopods, but they are particularly prevalent in lichens and bryophytes. Now, the designation of endophyte does not imply functionality. In some cases, the endophytism appears to be a common and regular part of their biology, but in other cases, it may just be an accident, or it may also be a strategy for a species to hang out until conditions are good for commencing their life cycle. So these are our results after putting all of Betsy and her collaborators' sequences into an LSU tree. We were able to parse them out by family, and I'd like you to notice three things. Number one, the endophyte sequences, which are in green, are spread across the entire class. However, they're conspicuously missing from the Helvelaceae here and the Tuberaceae here, and also the moss bryophytes, which are right here. So they're mainly most of the Pezizomycetes that are endophytes are mainly saprobic. At least that's what we are surmising. Some of the saprobe lineages like Plicaria and much of the Sarcomataceae appear to be widespread and common as endophytes. So there's something going on there that I don't really understand. And the third thing that I wanted you to notice, at least what I thought was striking, was that when I think about ways that fungi can get from one place to another, I now think about transporting plants by humans as being one of the ways that endophytes can get from anywhere in the world to wherever a human brings them. And now I want to tell you some stories from my life as a mycologist. This first one goes back to my time working on my doctoral degree at the University of Minnesota. This is Leticia Kumar, an undergrad who was helping out in our lab while I was there. She went on a truffling foray with me and discovered a new species of Hydnocystis. This was super exciting because it was the first time Hydnocystis could be documented from North America. And we thought it was going to be a simple thing to describe it and publish it before she graduated. And as icing on the cake, the new species cake, I collected a spore mat whose sequence matched the new species from the same woods. It was going to be great. Matt Smith and Jim Trappy gave us some specimens of related things to sequence and compare, and we borrowed others from herbaria, and then we analyzed our sequences. But it did not turn out to be so simple. By adding the sequences from North America into the tree, the genus became non-monophyletic, which to a fungal systematist is like fingernails scratching a chalkboard. So in order to fix the problem, we had to transfer Stephensia bombycina, this middle species here, which doesn't look anything like the Hydnocystis species on either side. So we had to transfer Stephensia bombycina to Hydnocystis. And we thought that took care of that. So we put more species, we did some more sequencing, borrowed more specimens, did more analysis. And this time we had some from South America. But when we did the analysis, we found that Hydnocystis singeri was making Paracotylis, which is both of these are, well, Paracotylis is a southern hemisphere genus. And Hydnocystis singeri had been described from Argentina. So that was a problem. So we had, but I was pretty convinced that it really was a Paracotylis when I compared the sequences between Paracotylis singeri, or what we transferred to Paracotylis, Hydnocystis singeri, the Hydnocystis, two species actually from South America, with Paracotylis pila from New Zealand. This whole part of this ITS sequence here is missing from all of the other Targetaceae. We call it an indel because we can't be certain whether it's an insertion or a deletion, but it is common only to these two groups of fungi. So looking at it morphologically, on the left we have Paracotylis, what we ended up calling Patagonica from Patagonia in South America, and Paracotylis pila with some podocarp fruits from New Zealand. And the reason I show the podocarp fruits is that New Zealand has no endemic mammals to disperse truffles, but we think that they are bird disseminated and maybe going after the brightly colored truffle, which also look like fruits. We don't know what the trophic level of the Paracotylis in South America is, however. So we did some nomenclatural rearrangements. And so Hydnocystis was all monophyletic. Paracotylis was monophyletic. We resurrected Densocarpa from Stephensia schnoori and made it Densocarpa. And so all was finally well. But by this time it was 2017. We published our revision of the truffle genera in the Targetaceae 8 years after Leticia's exciting discovery. And by this time I was working at the University of Florida and Leticia was ready to graduate with her PhD. So that was that story on how slow it can take to actually write a paper. And just to let you know, there are cup fungi in this group. They include Geopixus and Targeta. So there we have, now we know that Hydnocystis is a Northern Hemisphere the genus, and Paracaudelus is a Southern Hemisphere genus, except for when it gets transported to other places like England. And Densocarpa is in the Northern Hemisphere. As long as we are talking about the Southern Hemisphere, we can talk more about the fungal connections between Southern South America and Australasia. Don has been collecting in Patagonia for many years, like some of the famous mycologists also from Harvard, and such as Thaxter. And Matt Smith got a grant to study the fungi associated with the Nothofagaceae, which are called the Southern Beach, in the Patagonia region of South America. He wanted to test the hypothesis that fungi associated with particular hosts went with those hosts and continued to evolve following the breakup of Gondwana. The only native ectomycorrhizal trees in Patagonia are species of Nothofagaceae. On this graphic, you can see where fossil Nothofagaceae have been found in Antarctica and in Australia, all of these green dots, and where the present day Nothofagaceae exist. So they are in the Patagonia region of Southern South America and also in Australasia. Matt and his team found that this is the case in numerous examples. So I was lucky enough to get to do some truffling in Patagonia with Matt and his team. And this is just a picture of Patagonia with their Nothofagaceae forest. It's truly a beautiful place. But there are other trees there as well that are not ectomycorrhizal. They're sometimes known as the monkey puzzle tree, Araucaria. In some of the places we had companions helping us. But one of the things that I was in particular looking for, as were other members of our team, was Amylascus. And the reason is because Amylascus was described from Australia by Jim Trappy in 1971 as a truffle that has an amyloid ascus, which is unusual for truffles. Most truffles have lost the amyloid character. But he was able to describe two species from Australia. He gave Matt some collections that he and his team had made in 2001. And I was lucky enough to get to take on that particular project. So we were looking for Amylascus in South America because Jim had collected at least 3 or 4 species that were new in South America, but they didn't realize that that's what they were at the time, because the asci of the South American Amylascus did not turn blue in Melzers, and the spores were dark brown rather than yellow or colorless. We also found species that they had not collected that we've named Amylascus cinereaceus. And the interesting thing about this is that we also found mitotic spore mats that were not on the surface of the soil. They were buried in the soil. And we're not quite sure why that was. There are some birds that do some excavating for truffles there, and it could be that they ended up burying them, but we're really not sure. In the end, we were able to describe 5 new species from South America and a couple of new species from Australia. That's a paper that we'll be submitting soon. One of the take-home messages about this paper that I thought was interesting is that Amylascus, in writing this paper, we are going to be also redescribing what Amylascus is, the concept of Amylascus, because it's not just, it's got a much wider range of characteristics than the 2 that Jim had in hand at the time that he described the genus. And so you can understand that the first person to describe a rare fungus really doesn't have a chance of getting it completely right, because it can take decades to find enough relatives to get a more complete picture. Our collaborator, Camille Trong, who is now in Melbourne, Australia, did the lineage dating analysis on Amylascus, and a new lineage that we discovered down there too, that is sister to the Amylascus pachyplodes lineage, and only found in South America, at least so far. She found that the Pezizales had a mean age of 276 million years, that the clade of Luteoamylascus and pachyplodes and Amylascus has diverged about 28 million years ago. And that at that time, Antarctica had a warm temperate climate and is thought to have been covered by forests until about 25 million years ago. Okay, so now I'm going to shift focus to what our team is concentrating on now. And I'm delighted to have some Asheville people here because we did a lot of collecting in your area. We got a grant from the National Science Foundation to study the Pezizales, and specifically to generate genomes to use for phylogenetic analysis to stabilize the backbone of the phylogenetic tree that we have at the moment, so that we can have a better idea of how the species, genera, and families are related to one another. Our first official trip was to the Great Smoky Mountains National Park. It was our first official trip because we got the grant during the COVID lockdown. And so the trips that we took were unofficial, and we needed to finance them ourselves, but we did it. We also solicited collections from serious collectors from as many places in the US as possible, and even some from Canada and Europe. I've listed some of the most prolific collectors who have been sending us samples, and some even have been putting their samples into CTAB buffer for us and sending them along as well, and also have been making great documentation and photos of their collections, which has been really invaluable. The Pezizomycetes are known to fruit in slightly disturbed areas, such as along paths. This is a path in the upper Midwest woodland. I noted that somebody from Minnesota is here. This is in Minnesota. And here we have a lake. They're also great to find along lake banks. And they're great to find along creeks. In fact, if we had paid attention to that sign, we wouldn't have come across these gems: Scrutolinea, Adelphella, Pachyella. So those are just some of the places. And I didn't mention dung because they aren't very lovely to look at, but that's another great substrate that we often bring back from our forays. This is a picture of our website where we're posting some of our results. And our goal is to generate more genomes to hit at least 80 high coverage genomes across all 22 families. Presently, there are 43 genomes for 25 genera and 12 families, but we're generating about 20 at the moment, 20 extras. We also want to generate 300 low coverage genomes targeted at nodes on the phylogenetic tree that have low support in multi-gene treatments. So the low coverage genomes will give us a lot more information than like five gene treatment, phylogenetic treatments do at the moment. So there's 9 families that lack a genome right now, but I'm happy to say that we've been able to cover at least 8 of those. So the things in green are whole genomes. The things in blue are low coverage genomes. Now I just want to give a shout out to some of the people who have been working really hard on this project. Benjamin Lamond is at the University of Florida and is in Matt Smith's lab. He has been working on the low coverage genome assembly and also isotope analysis and doing a systematic look at the Morkellaceae truffles. You can see a couple of those truffles here. The reason the isotope analysis is important is because we have made assumptions about the trophic strategy of lots of species. We selected some of these to look more carefully at. The isotope signals from saprobes, ectomycorrhizal fungi, and parasites are different. So I'm really anxious to see the results. We don't have them yet. There we go. I'll get this before the end of the talk. Then in Greg Benito's lab up in Michigan State University is Judd Van Wyck. He's doing microbiome analysis of above ground and below ground Pezizales, as well as working on whole genome assemblies. The reason that the microbiome analysis is really interesting to me is that we know now that some of the truffle odors that are emitted to entice mammals to come and dig them up and eat them and eventually disperse the spores are in part caused by the bacteria that reside within those fruit bodies. And there is some evidence that the bacteria in the microbiome tends to be conserved within genera, but different between genera. So we're taking a closer look at that, as well as what the bacteria can tell us from the above ground relatives. The other thing that I'm quite excited about doing data dives into, or at least having the people who know more than me do data dives, is looking at the genomic information we can find about how apothecial modifications in truffles occurred. Here we have a couple of different truffles that look a bit the same, but they're not closely related at all. This is Hydnocystis and this is Genia. Hydnocystis is in the Tarsiataceae, Genia is in the Pyronemataceae. This cottony-like stuff right here are the tips of paraphyses that have overgrown the asci. Down here, you can see that the covering within this opening here is made up of the paraphyses tips that have intertwined together and produced a covering internally that is very similar to the external covering. I'm also particularly interested in finding out more from the genomes about the relationships and how they came about, how the modifications came about within Plycariella, which is we will be putting this in Pachyplodes. It is an above ground version of Pachyplodes. But this group is really interesting because you have the cup fungus. You also have a species that is kind of in between a cup fungus and a truffle. Here you can see that the truffle is completely solid, but here you can see, can at least imagine how the hymenium has folded over itself. We've also got lots of examples of above ground morels and many examples of truffles that are in the Morellaceae and also in the Dissinaceae like Gyrometra and Hydnotria. One of the really fascinating things also is that we'll have a low coverage genome of Nothojaphnia, which is the sister of tuber, and above ground cup fungus. It's only in the southern hemisphere. Tuber is only a truffle, and it's only in the northern hemisphere. So there's lots of interesting, intriguing questions that I'm hoping that our genomic work will help to answer. Last but not least is the acknowledgement page. it's not only to gratefully acknowledge our funding source, but most importantly, to acknowledge all of the great collectors who carefully documented their collections and went to the trouble and expense of sending them to us. These collections tell us a lot about distribution, molecular and morphological variability, and crucially, give us precious data regarding rare species. And if I left anybody off of this list, I am really sorry. It was an oversight, and please do let me know. And now I can take some questions.
Thank you, Roseanne. That was amazing. We have, well, I only see one question in the chat. I'm sure that people have other questions. Please raise your hand or unmute yourself if you have one. But first, Randy Strobel, would you unmute yourself and ask your question?
Yeah, thank you. This is the first time I've ever heard of spore mats, although I think I've probably seen them before. I wonder if you could describe those in a little more detail. And then I'm also wondering if they produce sexual or asexual spores.
That's a great question. Thank you, Randy. They produce asexual spores. Their spores are clones of themselves, of the hyphae that produce them. We're calling them spore mats because it's a descriptive term that might be a little bit easier to understand than calling them anamorphs, but that's what they are. They're asexual forms. Does that answer your question?
Yes, thank you.
And Jason Karakian, hello.
Jason, hi.
Hi, Roseanne. I have a question about, I remember in Don's lab, some students were doing germinating spores of Morkella from herbarium specimens.
Oh.
Sometimes as old, and Don, please correct me if I'm wrong, sometimes as old as like 10 years or more.
Wow.
So I'm wondering if you know if there's other groups in Pezizales that have that capability.
That is a great study. I don't know anything about that, and I think that that would be a great project for somebody.
Maybe I shouldn't have said anything because I don't know what the results are.
I love that idea.
It's stuck in my mind.
Yeah, Jason, I think you should go for it.
Oh, it's not my idea. I'm just curious about it.
It's a really fascinating thing, and it would be, it would be tempting to try to correlate what you get from an herbarium with what you might get out in the spore bank, but of course the conditions are so different that you couldn't really make a comparison. But it would be a start, I think, to find out how old from an herbarium specimen can you get germination of a spore.
Yeah, it was at least 10 years, if not longer. And we were using herbarium specimens that had been stored away. They weren't treated in any special way. In fact, they were probably mistreated. They were fumigated and frozen and all sorts of things. But it's an interesting thing. I've mentioned it to many people, and nobody really knows much about it.
I'm not surprised that the information is from your lab.
Does anyone else have any questions? I'm sure there must be some.
I'm glad that I explained it so well.
If you do have a question, feel free to unmute yourself or raise your hand. Or
I do have another question.
Please, Jason.
Jason,
Sorry.
yes.
So that the literature for identifying, we all want to go out into the field and collect these and identify them. There is some, what kind of literature can we go to, especially for Pezizales? what can we use for literature to identify our specimens?
So you're talking about the fruit body specimens or the
Yeah.
spore mat specimen.
No, I'm sorry. Yeah, fruit bodies. I'm
Yeah.
thinking about cup
Either
fungi.
one, it would be really nice to have something comprehensive. The Pezizales came out with a book recently, well, maybe 7 or 8 years ago, the Ascomycetes of North America. And that is the most recent, somewhat comprehensive book for North America that I know of. Does anybody, Don, do you know of any others?
No, I think that's it. I would just point out that Pezizales isn't what you think it is anymore. It's been split up into a lot of different genera, which in some ways makes it easier. But basically you're back to monographs and floras and that kind of thing to be able to do the identifications. They're not easy fungi.
No, and part of the problem is that just because something doesn't match a sequence on GenBank doesn't mean that it hasn't already been described. So you have to go, so if you do get something that you're not able to track down in the literature, it doesn't mean that it's new. So you have to track down type specimens, and Don has done probably the most work of anybody to study type specimens of the Pezizales. But you have to track them down and study them and compare them. And a lot of times the really old ones you're not going to be able to get a sequence from. So so at that point you have to do morphology and then try to recollect from the same geographic locality and hope that you can find that species again and get a sequence for it. So it's not as you saw from my talk about the paper with Leticia, dealing with a new or what you think is a new species is not it's not simple.
Thank you.
Rosanne, I wanted to congratulate you for a wonderful talk.
Hi, Teresa.
Hi. And also I wanted to ask you, how easy is it to cultivate from spore mats?
Oh, I have not had any luck cultivating from spore mats, but I haven't tried Morkella. Laurie Karras did a lot of work with the Morkellaceae and Dissinaceae spore mats and was able to get cultures from not from the spores, but from the hyphae that were underlying the sporophores. So she would take minute pins and grab one of those hyphae and put it in culture, and that's how she was getting those into culture. The things that I have been trying are ectomycorrhizal, and I suspect that that's why we haven't had any success with it. The Morkellaceae and Dissinaceae, a lot of them are not obligately ectomycorrhizal.
I was just rooting around for a paper. I think I sent it to you, Rosanne, of Morkella, and they were trying to germinate the spores from the spore mats, and their success rate was 1 in 100,000. So if you plated out 100,000 spores, you'd get one culture.
Wow.
So they're not functioning as we think of conidia or typical asexual spores. They're probably functioning in the sexual reproduction of these.
And this is why Don convinced us to not call them conidia, because we don't know their function is not as propagules. we think, but we haven't been able to prove yet that they may be, may have the other mating type, the opposite compatible mating type, and are able to produce or provide the nucleus to make a dikaryon in the fungi that are, or the Pezizomycetes that are outcrossing.
Rodney, you have a question? Would you like to unmute yourself and ask? Rodney?
Yeah, I'm on here.
Okay.
So I've seen cup fungi often holding water for a long time, and I was just wondering, like, does that attract insects possibly, potentially, and would that help with spore dispersal? And there was a hell of a Quiliatae that I've seen, like the elfin goblet that stands for weeks, and holds water for a long time. So I just was curious if there's some ecological role going on.
That's a great question, Rodney. Well, I don't know. I think that from what I've been able to see microscopically, Don, you'll have to help me out here, but it seems that there are that a lot of Pezizomycetes have an extracellular matrix that helps it to maybe take in water, but only up to a certain extent. So I'm not sure why you would want to, why a cup fungus would want to, would want to hold water. That would, I think, prevent it from being able to express its spores.
Yeah.
And might, I would think also.
Because the spore producing surface is more on the underside than in that cup inside. Maybe, I don't know.
No, the spore, no, this, which species are you talking about again? Helvella crowledii?
Helva, Queilitii.
It's a copulate Helvella.
Yeah.
And so the hymenium would be in the cup.
Okay. Well, I've just seen them. I saw one last year, and I was able to visit it like several times for like a month. It just held up for a long time.
That's a great question.
And I would see it like holding water, and I'm just like, what ecological role is this doing? Because this holds water for days, and I just wonder if it's some attractant, to a little puddle of water like that in the woods, in the forest.
I don't know, and I'm curious about what happens to it when it's mature. When the spores are mature, I wonder if it's still cup shape. Does it retain that cup shape the whole time, Don?
For a long time, and then it kind of basically withers away. I mean, it looked like a cup fungi stuck on a morel stem. Is what it looked like.
It does seem to me that it takes a longer time for the ascomycetes or the Pezizomycetes to mature than a mushroom.
Mm-hmm.
So it can take weeks.
Anyway, when I saw it just holding water like that, it just made cup fungi, they'll hold water for a little while, and I just thought, man, something like at least a mosquito could get in there and breed.
Yeah, well, that's a good question. I don't have a good answer. Don, do you have anything that you would answer?
No, I don't think so. We look at this as hydrating the fruiting structure and so forth, but I don't know that there's a function that I could come up with.
Anyway, one of those things I wondered when I'm out in the woods. Thanks so much.
Thanks for your question, Rodney. Mike Inskeep, you have a question. Would you like to unmute yourself?
Rosanne, Don complimented your ability to collect, and I was wondering what you attend to when you're out in the woods collecting.
So I look for places that have moisture. The Pezizomycetes that I'm aware of anyway tend to need a lot of water. So I look on soggy logs, I look under logs, I look at the base of, like, if it's truffles, I look for them in soil where water may have been caught, like catchments. I go slow. I don't cover much ground. Certainly not very fast.
Thanks, Mike. Eleanor Shavit, you have a question.
Yes, you mentioned that the bacteria has something to do with the aroma of truffles.
Yes.
So this is something that we have done quite a bit of work on, except I, for some reason, didn't know about the bacteria connection. So if you could expand a little bit about it, I'll be elated.
Well, I'm not the one who did the work, and I would direct you to Gregory Bonito. He
figured out he had to have a hand in it.
can you find his email address? I could try to find it for you.
No, I have it.
Okay, good. He is the master of bacteria within truffles, and it's his student who is doing the microbiome work on that, which I find fascinating, just as fascinating as you. And I'd also like to know more about it.
Thank you.
You're welcome. Good to see you.
Good to see you.
I have enjoyed your papers.
Thank you so much.
Thanks for your question, Eleanor. And Jason Tarakayan, you have another question.
Oh, it was more of like just a comment.
A comment, okay.
Yeah, I don't want to hog the whole thing,
Hey,
but
please share your comment with us.
Jason, I hardly ever get to talk to you. This is great.
hi.
Hi.
Yeah, no, I was just thinking about the puffing that you see in Pezizales,
Yeah.
or other cup fungi, and and I think it was Ingold who was talking about how that could be a kind of that mass discharge of spores may cause a kind of localized atmospheric, I don't know, disturbance or something that would help get the spores higher up into the atmosphere. Maybe I'm misremembering that.
No, you're probably remembering it much better than I am.
Well, yeah, so I wonder if that cup shape is kind of a way to, like a satellite dish. Like, I don't know how satellite dishes work, but if that kind of shape kind of helps direct that mass discharge into a column,
I suppose you could compare measurements among, because you have the cushion shaped ones like Ascobolus and Saccobolus, and then you've got sort of the platter shaped ones like Pulvinula, and then the cup shaped ones and the urn shaped ones. And I suppose that one could measure, how far up under certain conditions they're able to discharge their spores into the air.
Yeah,
I'm not aware of anybody who's done that. Dawn, do you know?
Yeah, Anne Pringle and her group did a study, and they were able to show that the discharge itself creates wind, and that wind in its turbulence carries the spores. So you can, Jason, you can find that. I don't have that in my head right now, but it's something about creating the wind. But it's based on fluid mechanics, basically. What happens when the air is when there's turbulent air through the discharge?
Okay, I'm glad I didn't make that up.
No, you didn't make it up. I don't think I don't remember Ingold talking about it as much, but yeah.
I might have gotten them confused.
There is that paper.
So if I can join to the conversation.
Hello.
Hi, Rosanne. I love a lot your talk.
Nice to
And
see you.
yeah, I would like to, because you are talking about the cup shape, at least what my impression is in Leotiomyces that are cup shaped is that when the weather is dry, they tend to close. So probably it's a way to protect the high minion. And then when the weather change and we have enough humidity, they open. And it's the moment that the acai shoots. So I think that's probably one of the explanation for the cap shape. I don't know if Don or Jason agree with that.
I don't know that I've
Difference-
observed.
Oh, sorry.
Again, we need to ask Don this.
No, you don't. But I was just going to say these are much fleshier and bigger than the fungi you work on, Luis. And I don't know of a lot of folding and cupping with humidity. And I think it has to do with the fleshiness of these fungi.
Okay.
But Rosanne?
But I love your talk, Rosanne. And Rosanne, and you show some slides at the end. And you talk about this paraphysis with this interesting morphology.
Yes.
So are they covering the menu? So they are above of the acai, I guess.
Yeah, so they overgrow the acai in some cases. I mean, it depends on the species, but there is one Amelascus I know of that makes paraphyses that end up covering the acai, but they don't really intertwine very much. Then there's another species where they do intertwine and they actually make what's called an epithecium that is similar to the accipulum.
And in those species, the acai shoot?
Oh, in those species that the acai do not shoot their spores. So those are truffles. And so they are depending on odors being transmitted above ground to attract mammals to come and eat them. And they're dispersed by mammals in their fecal matter. And it makes sense that this kind of an evolution would occur because they can be dispersed so much further away from where they were. But the question is, would they end up in the right habitat? But mammals tend to have tend to stay in the same kinds of habitats. So the chances are pretty good that they would end up in a habitat where they could then germinate, and in most cases, they would be needing to make a relationship with a compatible root to start growing.
Thank you.
Well, thank you so much, Rosanne, for a wonderful presentation. This presentation was recorded. A link to the recording is going to be distributed to all of the participating clubs. I know some people arrived late and would like to see the whole thing. So in the next week or so, a link will be going out to all the clubs. Rosanne, thank you so much.
It was my pleasure. Thank you. Thanks for being a great audience.
I look forward to seeing all of you in 2 weeks for a talk by Don Pfister. Until then, happy hunting, and look forward to seeing you all again soon. Bye.
Bye.